Yes.
Okay, great. Okay, thanks again for the introduction, and I'm really glad to have the opportunity to give a research talk at Shuimu BioSciences. And my name is Xiangyu Liu. I'm from the School of Pharmaceutical Sciences, Tsinghua University, and I'm going to share with you a few stories on the structural biology and drug innovation for the beta-adrenergic receptors. And I'd like to start my story by sharing a photo of myself, which was taken last year at March. That was the beginning of the COVID-19 pandemic. At that time, the students are stuck at home and there's nothing going on in the lab. So I decided that I would start running and to get some exercise. And once I started running, I bought a sport watch to monitor my heart rate all the time. And here's a 24 hours heart rate record of myself one day in March 2020. As you can see here, that day I wake up at around 7, then I walk to the lab, and then I sit in front of the computer for the entire day. I went for a running in the afternoon, and after dinner, I play with my son for about 2 hours, and then I sit in front of the computer again. So the lowest heart rate of that day is 40 times per minute when I was asleep. And the highest heart rate of that day is 160 times per minute when I was running. So it's amazing that our body can quickly sense the status of a movement, either as it sleep, sit, or running, and quickly adjust the heart rate. And this is done by different mechanisms, but one of the most important one is the sympathetic nervous system. It uses the adrenergic receptor to mediate so-called fight or flight response that regulates our heart rate, blood pressure, and cardiac contraction. In our heart, we have two important beta-adrenergic receptors, which are beta-1-AR and beta-2-AR. These two receptors are G-protein coupled receptors, which means when they are activated, they regulate the downstream signaling through the G-protein pathway. There are different kinds of G-protein, subtypes of G-protein in your body. And the beta-1-AR mediates through the GS, the stimulatory G-protein pathway, where the beta-2-AR coupled to both G-protein and GI, the inhibitory G-protein. Under normal physiological conditions, only beta-1-AR is selectively activated and regulates the heart cardiac regulation through the GS pathway. At this stage, beta-2-AR is not activated to avoid interfering with the beta-1-AR mediated cardiac regulation. However, under stress conditions, the overactivation of beta-1-AR is toxic. It may lead to cardiac apoptosis. So at this stage, beta-2-AR is also activated and mediates the cardiac protection function through the beta-2-AR GI pathway. So how could this happen? If we look at the endogenous agonist for the beta-adrenergic receptors, we have two important endogenous agonists, which are norepinephrine and epinephrine. So norepinephrine is a neurotransmitter released from the synapse, where epinephrine is a hormone released from the adrenal medulla. At resting state, the predominant endogenous agonist is norepinephrine. Where under stress conditions, epinephrine gets released and become the predominant endogenous agonist. So the key to the well function of sympathetic nervous system at resting state is that norepinephrine is beta-1-AR selective, as you can see here. It has a 10-fold higher affinity for the beta-1-AR compared to the beta-2-AR, where epinephrine is not selective. It has similar affinity to beta-1 and beta-2. This phenomenon has been known for decades and has been a puzzle for decades. Because if we look at the structure of norepinephrine and epinephrine, they are very similar. There's only one methyl group difference. And if we look at the structure of beta-1-AR and beta-2-AR, so here shows the structure of beta-2-AR. If a residue is identical between beta-1 and beta-2, we call it blue. And if it's different, we call it yellow. As you can see, the orthosteric ligand binding pocket, where norepinephrine and epinephrine binds to, are identical between beta-1 and beta-2. So if the affinity difference is not contributed by the binding pocket, what could it be? Could be the binding pathway kinetics, because we know the binding affinity is contributed by the association rate and dissociation rate. To check this, we performed binding kinetic study of norepinephrine towards the beta-1-AR and beta-2-AR. And as you can see here, norepinephrine have similar dissociation rate to these two receptors, but has a 20 times faster association rate towards the beta-1-AR compared to the beta-2-AR. That is to say, the association rate determines the selectivity of the norepinephrine towards the beta-1-AR. Which part of the receptor contributes to the faster association rate? Could be the extracellular part or the intracellular part, because we know the extracellular part of the receptor is a binding pathway of the ligand to the pocket, where the intracellular part determines the dynamics of the receptor between inactive state and active state. To confirm this, we generated two chimerical receptors, one with the extracellular part of beta-2 and intracellular part of beta-1. The other with the extracellular part of beta-1 and intracellular part of beta-2. And then we perform binding kinetic studies again. As you can see, the chimerical receptor with the extracellular part of beta-1 shows much higher, faster association rate and higher affinity to norepinephrine. That is to say, the extracellular part of the receptor determines the fast association rate of norepinephrine and thus the high affinity. And what is the detailed mechanism? To check that, we need the high resolution structure of the human beta-1-AR. However, despite human beta-1-AR being such an important drug target, there is no high resolution structure of this receptor. It's because the very similar human beta-2-AR is one of the best studied GPCRs. And this is mainly done by Kubiak Lab and collaborators. Here shows a few examples of the beta-2-AR in either inactive states or active states. So actually, before the publication of our work, there are about 35 structure entries of human beta-2-AR in the PDB. Furthermore, a homolog of beta-1-AR, the target beta-1-AR, is another very well studied GPCR, and this is mainly done Chris Tate's group and other labs. And here show a few examples of target beta-1-AR, and there are again around 30 structure entries in the PDB. But as I said before, the human beta-1-AR and beta-2-AR have different extracellular surface and different affinity towards norepinephrine. The target beta-1-AR and human beta-1-AR also have different extracellular surface and different affinity towards the norepinephrine and epinephrine. Furthermore, despite we have like over 60 structure entries of human beta-2-AR and the target beta-1-AR, there's no structure of adrenergic receptor bound with norepinephrine. So we don't know if norepinephrine binds the receptor, we could see some side chain rearrangement. So with this information, we decided we want to solve the structure of human beta-1-AR. And after solving some technical issues, we succeeded in solving the structure of human beta-1-AR bound with antagonist Carzolo in inactive conformation, and human beta-1-AR bound with high affinity agonist BI or endogenous agonist epinephrine and norepinephrine at active confirmation. Once we have the structure, we look at the ligand binding pocket. As we predicted, they're very similar between beta-1-AR and beta-2-AR. So the absence of the methyl group in norepinephrine did not induce any side chain rearrangement. So the binding pocket difference, binding pocket does not explain the selectivity of norepinephrine towards the beta-1-AR. But if we look at the extracellular surface of the receptors, we can see they're quite similar between beta-1 and beta-2, either in inactive conformation or inactive conformation. With the high resolution structural information we got, our collaborator Jonas at Peter Gomez's group at Germany performed metadynamic simulations to find out how norepinephrine binds to the receptor. The result is very interesting. He found that norepinephrine first contacts beta-1-AR and beta-2-AR through two negatively charged residues. In beta-1-AR, is these two green residues, two aspartic acid colored in green, and in beta-2-AR is this aspartic acid and glutamate. After the first interaction, the ligand would move inward, further inward, and inward. And then at this stage, those ligands need to go through an aromatic gate formed by two aromatic residues. In beta-1-AR is this two phenylalanine, while in beta-2-AR is this phenylalanine and this tyrosine. And after that, the ligand binds to the final position. So if we check the binding pathway, we could see there are 6 residues different between beta-1-AR and beta-2-AR. And if we switch these 6 residues, the residue color yellow here, in beta-1 and beta-2, we could decrease beta-1's affinity to epinephrine and increase its affinity to beta-2. Increase beta-2's affinity to norepinephrine. We further show that the mutation mainly affects the association rate of the ligand to the receptor. So how about epinephrine? Jonas also performed metadynamic simulations of epinephrine bound to the receptor, and we found that epinephrine is very similar to norepinephrine. That is to say, it also adopts different binding pathway in beta-1-AR and beta-2-AR. So how could norepinephrine be in beta-1 selective where epinephrine is not? We take a close look at the structure and we found that because the addition of this methyl group, it changes the electric distribution, and as a result, this nitrogen is less positively charged in epinephrine compared to norepinephrine. And then we check the natural distribution of the binding pathway. We found that in beta-1-AR, the binding pathway is a continuous negatively charged cavity, while in beta-2-AR is composed of 2 negatively charged regions linked by a neutral gap. So we guess that we propose that maybe because of the charge difference, epinephrine does not have a preference over these 2 binding pathway, where norepinephrine has a preference towards this one. Indeed, if we plot the free energy profile of the binding events, we could see norepinephrine shows similar free energy profile in beta-1-AR and beta-2-AR, where norepinephrine shows different behavior. And we also notice that norepinephrine seems to be trapped in a no minima position, pointed here by the arrow, and we found that is this position colored in gray. If we perform an unbiased simulation to this binding pose is stable for like 1 microsecond. So it's a pretty stable binding pose. We then claim that because norepinephrine gets trapped in this position before it can get to the final position, that slows the association rate of norepinephrine to beta-2-AR, and as a result, it has a lower affinity to beta-2-ER. This work was published last year on Cell Research. And how can this structural work guide the drug development? More specifically, how could this structural work guide the development of subtype sedative drugs? Sorry. So as I just said, the ligand adopt two different binding pathways to bind to the receptor. And we then think, what if we richify the conformation of the ligand? Because along the different binding pathways, the ligand may need to adopt different conformations. Furthermore, during a study, we noticed that a special residue, this phenylene 45.52. So this 45.52 means is a special labeling system in GBCR, which means this phenylene is a residue in ECL2 and is relatively conserved in GBCRs. So this residue is phenylene in both beta-1 and beta-2, but the environmental is quite different in these two receptors. In beta-2-ER, it's surrounded by 4 aromatic residues, but none of these 4 residues are the same in beta-1-ER. So then we think, what if we enhance the interaction of the ligand to this residue? Oh, furthermore, we show that if we mutate this phenylene to alanine, it has different effects in beta-1 and beta-2, as you can see here. The mutation has a large effect in reducing epinephrine or norepinephrine's affinity to beta-1, but has a smaller effect to beta-2. So by combining this, first, we want to richify the ligand, and second, we want to enhance the ligand's interaction with this functionally different residue, phenylene. We decide to make some compounds like this. By doing this, we richify the conformation of the ligand, and we have closer interaction with phenylene. But to do that, actually there are 8 possible isomers because of different carrier center. And our collaborator Peter Gomez's group synthesized all of the isomers and characterized all of them. Here's the result. So as I said before, epinephrine is not sedative in beta-1 and beta-2. Well, norepinephrine is sedative towards the beta-1-ER. One of the isomers, we call it super epi or super norepi, becomes beta-2 sedative. So it not only reduces the affinity to beta-1-ER, but also the efficacy. So as you can see, it shows reduced Emax. Actually, super epi is about 400-fold sedative towards the beta-2-ER. For comparison, previous the most sedative beta-2 agonist is Saventor. It's a compound that is clinically used to treat asthma. And Saventor has about 1,500-fold selectivity towards beta-2-ER. But it achieves the affinity by extending this long tail of the compound to the next conserved extracellular pocket. Well, super epi achieves similar affinity by adding just 2 carbons. As you can imagine, we could add the tail of Saventor to this to the super epi scaffold and to obtain a small molecule with even higher selectivity. Actually, our collaborator synthesized this compound, and in our hand, this compound showed like 10,000-fold selectivity, which is the most selective beta-2-ER agonist I have ever seen. So this phenomenon is very interesting, and we want to know the mechanism. We saw the structure of super epi bound with beta-2-ER, and indeed, super epi has strong interaction with this phenylene 45.52. Actually, this residue needs to move upward for about 1 astron to accommodate super epi. Otherwise, super epi binds to very almost identical position as epi. when we performed MD simulations, we could see that super epi binding pose is very stable in the 2AR. But if we just pose super epi in beta-1AR and perform the simulation, the binding pose is not stable. After 1.5 microseconds, the ligand would fly out, and the hydrogen bond between the ligand and TM5 just disrupted. So this phenomenon suggests that unlike the selectivity mechanism of norepinephrine to beta-1AR, which is contributed by the binding pathway, the super epi selectivity is a different story. Is because of a less stable binding pose in the beta-1AR. As I said before, the phenylene 45.52 has different environments in beta-1AR and beta-2AR. So if we switch these 4 residues between beta-1 and beta-2, we could decrease beta-2's affinity to super epi and increase beta-1's affinity to super epi. And if we mutate these four residues, super epi binding pose would be stable in the beta-1AR with these 4 mutations. That is to say, the different interaction with the phenylene 45.52 is a key to the super epi selectivity. A few years ago, our lab has published a work on the structure guided development of M3 receptor selective antagonist based on a single amino acid difference between the M2 receptor and M3 receptor. So M2 muscarinic receptor and M3 muscarinic receptor are very similar, except in the negative binding pocket, they have one residue different. Based on this single amino acid difference, we modified a ligand starting from a non-selective ligand. We ended up with a ligand which shows about 100-fold selectivity towards M3 receptor in binding assay and more than 1,000-fold selectivity in animal studies. We further confirmed that the molecular mechanism of the selectivity is just like what we designed. We use a halogen atom, in this case is a fluoride, to point towards a leucine residue in M3. And if we place the compound in M2, the fluoride would clash with a larger residue, the phenylene in M2. So in this case, we further show that even for beta-1AR and beta-2AR, which have identical residues in the ligand binding pocket, some of the residue, for example, in this case is phenylene 45.52, may have different function or different interaction with the ligand because it has different environment, and we can again design selective drugs targeting on these residues. So to conclude this, I would say that it's possible to develop subtype selective drugs for GPCRs with identical allosteric pockets by carefully exploring the environment difference around the allosteric pockets. And I would claim that high resolution structures will provide valuable information to guide the research. So I just introduced a story on how challenging it is to develop subtype selective drugs. As I showed before, the challenge is mainly because the allosteric ligand binding pockets are very conserved in different types of GPCRs. But if we look at the surface of the receptor, we could see that there are some areas that are quite different between, for example, beta-1 and beta-2. So is it possible if we find some ligand or some small molecule that bind to the less conserved region of the receptor and modulates the function of the receptor? So here comes the idea of the allosteric drugs, or allosteric modulators. So allosteric modulators are small molecule or drugs that bind to any place on the receptor that is away from the allosteric ligand binding pocket. And allosteric ligand binding pocket is where the endogenous agonist binds to. And the first allosteric modulator I would like to introduce is compound 15, which is the first negative allosteric modulator for the beta-2AR. And this compound is found through a DNA encoding library screening. This work was done by collaborators Bob Nefku's group and Professor Chen Xin's group. Basically, what they did is that they use a DNA encoding library, which is composed of millions of compounds, which are unique DNA tag linked to each compound, and then they performed affinity selection to the receptor, and by sequencing the bound compound, they could get the structure of the compound. They screened 190 million compounds and resynthesized 16 compounds for the studies. Of these 16 compounds, they found compound 15 seems to be the most interesting one. So here's shows the structure of compound 15. This compound can redshift the agonist competition curve. That is to say, it can decrease the agonist affinity, and it has around 1 micromolar affinity to the beta-2AR. Of this result, the most interesting result comes from panel E. So here shows the interaction between the receptor and the nanobody 660, which recognizes the inactive conformation of beta-2AR. So if you add a high affinity agonist BI, you could see decrease in interaction. And if you show add antagonist ICI, you get a good interaction. Compound 15, as we said before, it stabilizes the inactive conformation of beta-2AR, but it also disrupts the interaction between beta-2AR and compound and nanobody 60. That is to say, compound 15 most likely bind to the intracellular part of beta-2AR and modulates the receptor function, which was very interesting because at that stage, no such compound has been reported before. So we want to know the mechanism. I got the compound. I tried to crystallize the receptor with this compound, and we connected a 2.5 angstrom data set. From the data set, indeed, I saw some weak density at the intracellular part of the receptor, and the density is slightly stronger compared to the noise in the control data set, but it's still very weak that I could not confidently build the model just based on the weak density. So here shows two examples of how I model the compound, but none of them are correct in the end. So from this result, we conclude two things. First, compound 15 indeed bind to the intracellular side. Second, the density is very poor, so because we don't have enough compound 15 occupying the crystal, and we call it poor occupancy. And the occupancy is poor because compound 15 has poor solubility. So we want to increase the solubility of the compound. At this stage, our collaborator, Professor Chen Xin at Changzhou University, synthesized this derivative of compound 15. Basically, he added this tail we call polyethylene glycol carboxyl group to compound 15. And we know we could add something as this position because this tail was used to couple to the DNA tag. The new compound, we call it compound 15PA. And this compound shows similar activity, but much more soluble compared to compound 15. So with this new compound, I got another 2.7 angstrom data set, and in this data set, we clearly see the density for compound 15. Indeed, compound 15 binds to the intracellular side of the receptor, and it binds to the intracellular of TIM1, TIM2, TIM6, and TIM7. So how does it work? So in GPCR, we know that upon the activation of GPCR, TM6 need to move outward. So if we compare a inactive conformation of beta-2-ER and the active conformation of beta-2-ER, we see the largest difference come from the intracellular part of TIM6. TIM6 need to move outward for like 14 angstrom. So compound 15 binds in between TIM6 and TIM7. Actually, it induced a new cell bridge between TM6 and TM7. So it basically locked TM6 in inactive conformation. Indeed, if we perform MBT simulations, we could see the distance between TM2 and TM6 is like 3 angstrom shorter in presence of compound 15 compared to the control structure. So this is the first mechanism. Compound 15 binds between TM6 and TM7 and knock TM6 in inactive conformation. And that's why, even though compound 15 binds to the intracellular side, it can allostorically modulate the conformation of the receptor and decrease agonist affinity, which bind to the extracellular pocket. Furthermore, compound 15 binding pocket is overlapping with the G protein or arrestin binding pocket. So that's the second mechanism. Compound 15 can directly compete with transducers like G protein or arrestin and interfere with signal transduction. There are 21 residues in beta-2-ER that forms compound 15 binding pockets, and 2 of them are different. And because of these 2 different residues, compound 15 can decrease agonist affinity in beta-2-ER, but not in beta-1-ER. And compound 15 binding pocket might be a general allosteric pocket in other GPCRs, because slightly before the publication of our work, there are 2 papers published on Nature showing the similar binding allosteric modulation pockets in CCR2 and CCR9. And the second allosteric modulator I want to introduce is compound 6, which is the first positive allosteric modulator for the beta-2-ER. And compound 6 comes from the same DNA encoded library screening, but it has different properties compared to compound 15. It can left shift the agonist competition curve, which means it can increase agonist affinity, and it can cooperate with either G protein or arrestin or a nanobody called nanobody 69. Sorry. So nanobody 69 is very important because we use this nanobody to stabilize the receptor in active conformation in Christopher work. So here shows the structure of compound 6. We have been struggling to get the compound 6 binding pocket by structure studies for years, and we found the same problem. We could not see the density of compound 6. It has very similar problem with compound 15, that the compound is just not soluble. As you can see, in the crystal condition, it just precipitates. But during study, I found that one of the precipitant we use in crystallography called PEG 400 can actually increase the solubility of the compound. So I come up with an idea to explain that. I need to briefly introduce how we perform the crystallography work. We use a method called lipidic cubic phase crystallization method, LCP, to do the work. Basically, we purify the protein, and here we color it blue for clarity, and we mix it with the lipid to form the LCP, and then we set up the crystals. If your protein is stable and if your condition is correct, you will get nice looking crystals and ready to collect data. So there are 2 commonly used host lipid for LCT. One is called MEG 7.7 and one is MEG 9.9. And 7.7 has a shorter hydrophobic tail and 9.9 has a longer hydrophobic tail. And because of the longer hydrophobic tail, the MEG 9.9 forms thicker lipid bilayer, and we notice that the crystals grow in presence of MEG 9.9 often requires higher precipitate condition, and in our case, is higher PEG 400 condition. So all previous crystals are grown in using MEG 7.7 as a host lipid, and the crystal condition contains 25% PEG 400, as you can see here. Compound 6 is not soluble in this condition. Then I changed the host lipid to MEG 9.9 and screened for new conditions, and we found a condition contains 45% PEG 400, and in this condition, compound 6 is fully soluble. With this new condition, I was able to find the density for compound 6. It binds to a very interesting position called ICL2. Here shows the overall structure. As you can see, we have active confirmation of beta-2-ER. We have the high affinity agonist BI, and we have the nanobody 6B9 to stabilize the receptor in active confirmation, and we have compound 6 band just above SL2. So why SL2 is interesting? Because SL2 is a loop in inactive beta-2-ER, and it's a short alpha helix in active beta-2-ER. The alpha helical conformation of the SL2 places this phytoadenine 139, in a proper orientation to interact with G protein. Compound 6 just band right above SL2 and stabilize the alpha helical conformation of SL2. Also, the binding of compound 6 requires about 3 angstrom inward movement of TM3, where the inward movement of TM3 would push TM5 and TM6 to move outward. As I said before, the outward movement of TM6 is a hallmark of GPC of beta-2-ER activation. So that's why, again, even though compound 6 binds to the intracellular side, it can allosteric enhance the agonist affinity, which binds to the extracellular side. So compound 6 binding pockets contains 14 amino acids, and 7 of them are different between beta-1 and beta-2. As a result, compound 6 show pan activity to beta-2-ER and very little activity to beta-1-ER. But if we mutate the different residues, it can also work for beta-1-ER. So the first positive allosteric modulator we found in GPCR is M2 muscarinic receptor as pan, this ILY compound. This compound binds to the extracellular pocket of the M2 receptor and slows the dissociation of the agonist. Well, compound 6 is like the second pan we found for GPCRs, and it binds to a different pocket and have a very different mechanism. So to summarize my talk, I introduced 4 stories today. The first one is that we found norepinephrine takes different binding pathway to human beta-1-ER and beta-2-ER, and the binding pathway difference contributes to its selectivity towards the human beta-1-ER. Then we show that for beta-1-ER and beta-2-ER that have identical allosteric negative binding pockets, some of the residue, like the phytoadenine 45.52, may have different function because it has different environments, and we can target in on this residue and develop subtype selective drugs. I further show that this aside from the allosteric ligand, we could try to develop other stereo ligands that bind to the next conserved regions of the receptor and modulate the GPCR function. So with this, I would like to finish my talk and acknowledge my collaborators. And this work was mainly done when I was a postdoc in Brian's group. So I would like to first thank Brian Thompson, and I presented some crystallographic work done by Xinyu and Hong Tao. And I thanks to collaborator Ron Zhou and Bill Weiss at Stanford University, Brian Shurkis at UCSF, and Peter Gomez's group at FAU Germany, Richard Sanha's group at UCSD, Professor Chen Xin's group at Chengzhou University, and Bob Nefku's group at Duke University. And I thank Coolio and Spring 8 for help with automatic data connection. And thank you very much for your attention, and I'm happy to take questions. Thank you.
Okay, thank you, Xiangyu, for your informative presentation. And I would like to encourage our audience to submit your question using the Q&A box at the bottom of your screen. I will start with this question. I think compound 15 and compound 6 are isolated from the DNA encoded small molecule library. So, allosteric modulator are the hot field in the GPCR drug discovery, as they have a better selectivity, right? And the intracellular allosteric modulator is a promising mechanism for the drug discovery, especially for the small molecule. So I'm wondering, you select selective 16 compound for further study. Is there any compound are found among them binding to the extracellular side?
Well, for this beta-2 screening, I'm not sure actually. It's just by chance that we somehow find the intracellular binder. But you know, there are other GPCRs that contain extracellular allosteric modulators, and actually right now there are more and more GPCR allosteric modulated complex structures being solved. So I guess, I mean, the DNA encoding library screening is a nice way to screen for allosteric modulators, because you can block the allosteric pocket with a really high affinity ligands and screen for other binders. But besides that, if we have a structure, for example, if we have a structure of beta, let's say M2 receptor with an extracellular modulator, and we want to find a allosteric modulator for a very similar GPCR, we could just use in silico screening to screen for ligands that bind to a similar pocket. I guess that's another way to find allosteric modulators.
Yeah, great, fantastic. Thank you, Xiangyu. One more question, and so they are interested in compound 50 and compound 6, and they are isolated from the DNA encoded small molecule library. So is there any compound could be found in nature with the same or similar function as a compound 15 or 6 you mentioned in your presentation?
Oh, that's a very good question. I really want to know, but so far there's no reported such compound for beta-2 for sure. I just wonder if there's any similar compound for other GPCRs. Not that I know of, but I mean, I guess there's something. It's just we haven't found them yet.
Great. Okay. One more question. Have you or your colleague tried to optimize the compound 6 FA?
Yes. 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 Well, it's mainly our collaborators. They are working on that. that is a very interesting compound, and we for sure want to explore more possibility.
Great. I think we got time for 1 or 2 more questions. So please submit your questions using the Q&A box at the bottom. Okay. So if there is no more questions, I think it's a great segue to thank everyone for being here today. And I would really like to encourage you to contact us, Shuimu BioSciences, have a further discussion to see what we can help with your project. And you can also expect a record for this video to share with your colleague at any time. And feel free to ask any questions anytime, and we hope to work with you in the near future. And so thanks again, everyone, and have a great day. Thank you, Xiangyu.